Hello everyone, my name is Charu Saini and I am forensic biology educator on Think Saksha. In this video, we will continue the topic biological evidence part 2, that is blood stain pattern analysis. Blood stain pattern analysis is the second subtopic in biological evidence. So let's understand what is blood stain pattern analysis. So for that, we have to understand what is blood stain. Blood stain is simply a mark or a discoloration of blood on any kind of surface. So the blood stain pattern analysis is the application of scientific knowledge for the examination and interpretation of blood stains. So what is the scientific knowledge? These are the already developed scientific methods which we will employ to examine the blood stain we have found on the crime scene. And how and what we can interpret from this examination? First is morphology. We can know about the morphology of the blood stain, that is the shape of the blood stain. As we can see in this image, there are different shapes of the blood stains present. First, we can see a spherical shape of a blood stain. There are spiny edges coming out of the blood spots, blood droplets, and small droplets can be seen with irregular shapes. So second is the sequence. Sequence is the order in which these stains were created on the crime scene. From this we will get to know about the events, or about the occurrence of the events that were carried on the crime scene, like how the crime occurred with the sequence. If first this stain was created, then the first here the scenario happened, then later if we get the stains here, then the body or the victim was shifted from here to here. This is the sequence. And third is the distribution of blood stains, like how these stains are spread on the surface. The stains spread on the surface differ with the kind of surface. Suppose we have a smooth surface, then a proper spherical shape droplet will be created there. And if we have a porous surface like soil or concrete based material, then it will absorb the blood droplet, then irregular shape will be there. So the next is, okay, we know what is blood stain pattern analysis. Now what we will get to know from the blood stain pattern analysis, that is the sequence of events, like how the sequence was carried on the crime scene, the approximate blood source location, where like approximately we can get the idea that yeah, blood source was present here. What is blood source? It is the source from where the blood was coming. So we can get a rough estimate idea that yeah, blood source was at this distance from the blood stain pattern. Third is the position of the victim. Yes, we can know where the victim was present, was standing, sitting, whatever. The position, the intensity, and the number of impacts applied to the blood source. The position of the blood source, yes, we can get to know that. The intensity, from what intensity the blood source was hit, and the number of impacts. This also depends on the kind of weapon used to hit the victim. Fifth is the manner of death and can distinguish between accidents, homicides, and suicides. Blood stain pattern analysis will help us determine that whether it was an accident, homicide, or a suicide. Next, in this image, we can clearly see a crime scene, distorted crime scene where a lot of blood stain is present. So let's zoom it. Here we can see tag number 1, and here some kind of shoe prints can also be seen, I think, yes. And these stains are a kind of dragging. They're a kind of dragging. The direction of dragging is from here to here. And in this, here, blood stain is again present. Here also, everywhere blood is present. So from the analysis of these blood stains, what we will get to know that where the victim was present, what was the direction, and like what kind of weapon was used. This we can also get to know. Next, this is the another crime scene where we can see the blood stain patterns on the wall. There are different kinds of blood stain patterns, and a forensic expert is documenting the crime scene. From the study of these kind of blood stain patterns, we can analyze what was the situation on the crime scene. Now let's understand what is blood. So blood is a fluid which is 5 times more viscous than water, and an average adult has a blood volume of approximately 8% of his or her own body weight. So blood is divided into 2 parts. First is liquid portion, that is also called plasma. It is the liquid portion. And second is the cellular portion that contains all types, that is composed of all types of cells. First, red blood cells, that are also called erythrocytes. White blood cells, also known as leukocytes. These cells are divided again into agranulocytes and granulocytes. As the name suggests, granulocytes contains granules and agranulocytes does not contain any granules. Agranulocytes are again divided into lymphocytes and monocytes, and granulocytes are divided into neutrophils, eosinophils and basophils. Then the third part is platelets. These are also known as thrombocytes. So plasma contains water, proteins and other solutes such as inorganic salts and organic substances. So this is the composition of blood and you should remember it. Next we have formation of blood stains, like how blood stains are formed. We know blood is leaked, like blood falls from our body, but how it comes in a spherical form, how these shapes are formed, these spiny edges, how they are created. So this is created by the influence of viscosity, surface tension, cohesion force and gravity. As we can see, there are here 3 surfaces are present. This is a smooth surface and a scale is also in the photograph we can see. And yes, you guys should remember that scales are important during documentation. Measurements should be there, because in a simple photograph we will not get to know that what was the measurement of the stain or any evidence which was present on the crime scene. So scales are present. It is a kind of Abiafu scale. So first surface is a smooth surface where a spherical blood droplet shape is formed. In second, it is a cardboard surface on which spiny edges can be seen, and third is a thin paper on which we can see the blood was kind of absorbed in the thin paper. So let's understand what is viscosity. It is the fluidy nature of the blood. Second is surface tension. So the surface tension is created due to cohesion force. So what is cohesion force? If these are the particles of the blood, then these particles of the blood try to attract towards each other. This is the tendency of similar blood particles, that they attract towards each other and they tend to be towards each other in a spherical shape, and this creates a surface tension on the surface, which is a surface phenomena, and this creates a surface tension. Thus this helps the blood droplet to be in a spherical shape. In this image we can clearly see it is a blood source from which blood is leaking out, and when it leaks out it creates this phenomena. These are the particles of the blood, okay, that attract towards each other. Let me zoom it for you. See, these are the particles of the blood that attract towards each other and bind with each other. okay, because these are similar particles. This creates a surface phenomena, that is surface tension, and this does not let the blood come like this. That's why it is in a round shape due to surface tension and cohesion force. And the another two forces that are applied on a blood droplet are the air resistance and gravity. Gravity is applied downwards and air resistance is applied upwards. This creates a spherical shape of a blood droplet, and when it falls on any surface, it is round in shape. So this is how blood droplets are formed on any surface, and like this blood stains are formed. Okay, so now we know that blood stains are present, but to confirm whether these blood stains are blood only and not any other red liquid or blood color red liquid, to confirm that we will have to carry some chemical reagent test. This is called chemical enhancement. The chemical reagents that are used to identify the blood stains on any crime scene are called chemical enhancement tests. So the common chemical reagents are luminol, phenolphthalein, leucomalachite green, and tetramethylbenzidine. Luminol is used for latent blood stains. What are latent blood stains? Latent blood stains are the blood stains that are not visible by naked eye. Latent prints are any kind of print that are not visible by naked eye. We cannot see them. So in case of blood stains that were present but were washed by the perpetrator or the accused person to hide the evidence are called latent prints of blood stain. And luminol is the solution that can detect these blood stains even after they are washed. Phenolphthalein, leucomalachite green, and tetramethylbenzidine are the reagents that are used for presumptive testing of blood stain. Documentation of blood stain evidence is also very important, so we can analyze the photographs, the documented videography, notes taking and sketching. These all can be analyzed afterwards to reconstruct the crime scene, because crime scene reconstruction tells us about the events of the crime scene, like what happened there. So we need these blood stain evidences to be documented in the form of photography. Photography can be done in three types. First is from the far range, second mid range, and third is zoom range, like close range, very close range. Suppose these are this is blue color, but suppose these are the so many blood stains. So if we need to do the photography for the far range, we will cover this whole blood stain pattern. If we need to do the photography of mid range, then we will cover this much. If you can see the green color, please distinguish it, this much. And if we need to do the close range photography, we will consider only 1 to 2 drops in that area. And yes, scales are important. Adhesive scales are there that stick on walls, and photography can be done easily. So here we have a picture of hemoglobin structure. And why do I have it here? Because hemoglobin is present in blood and it contains iron. Iron is used in all of these tests as a catalyst. Let's know how. So these all tests are based on the peroxidase-like activity of hemoglobin. What? So these all tests are based on peroxidase-like activity. Remember this term, peroxidase-like activity. Hope you understand this. So what is peroxidase like activity? It is the activity of hemoglobin. As it contains iron. Iron in heme catalyzes the breakdown of hydrogen peroxide, which is added in this test. Okay, hydrogen peroxide is catalyzed by the iron which is present in hemoglobin, heme. Then hydrogen peroxide, it breaks down, and hydrogen peroxide further oxidizes the test reagent, such as in this test luminol reagent. Hydrogen peroxide will oxidize the luminol reagent and it will show us the bluish glowing color, as we can see in the image. In this image, first we cannot see any of the blood stain pattern, like no one can say that yeah, blood was here. It is clearly washed, but with the help of marvelous reagent luminol, if we spray it here, we can clearly see the glowing blue color, that yes, blood was present here, and it is due to the heme group which is present in the blood. So, luminol reagent contains 5-amino-2,3-dihydro-1,4-phthalazine-dione. You have to remember this. And here also it's written, in the presence of hemoglobin, luminol gets oxidized and produces a blue glow light emission. It should be luminol, guys. Luminol. It's O here. Okay, and here oxidizing agent is hydrogen peroxide. And what is Fe2 positive, that is iron from the heme group, doing here? It catalyzes the hydrogen peroxide. It breaks down the hydrogen peroxide to do the oxidation of luminol. Next we have leucomalachite green. In this test also same thing happens. We have to collect the suspected stain on a swab and we react it with leucomalachite green reagent, and after that we add 1 to 2 drops of 3% H2O2. A blue green color is a positive indicator of blood. In this test also similar thing happens, that is when the Fe present in heme group of hemoglobin present in blood, it breakdowns the hydrogen peroxide. Hydrogen peroxide then oxidizes the leucomalachite green to show blue color. Next, in phenolphthalein test, Castle-Meyer test. In this test, similar thing happens, and phenolphthalein which is colorless, it oxidizes into phenolphthalein which is bright pink in color, and the blood stain can be identified. Tetramethylbenzidine TMB test. The similar thing happens. TMB is a chromogenic substrate, that is, it shows color, and in the presence of hemoglobin's peroxidase activity and H2O2, it gets oxidized and produces a blue-green color. So, in this video, we have discussed what is bloodstain pattern analysis and what it tells about. It tells about the sequence of the events that has happened on the crime scene, manner of the death, position of the victim, and intensity and the number of impact of the blood source on the blood source. What was the intensity to hit on a blood source? This all we get to know from the blood stain pattern analysis. Yes, we also discussed the color test for identification of blood-stained blood on the crime scene. And in the next video, we will discuss about how blood stain patterns are analyzed and their types, because their types are important for your syllabus and examinations such as UGC NET and FACT. So that topic is important. Do watch the next video also. Thank you.